Today's topic is structure of chromosomes for class 10. You learned in class 9 about the cells, about the components or contents of the cell, about the cellular organelles present within the cell. Now, you know the brain of the cell is the nucleus. Now, here I am showing an animal cell. This is the cell membrane. This is the centrosome, which is the specific structure for the animal cell, which is responsible for the cell division. Means they form a spindle-like structure in time of the cell division. In after 2-3 classes, after 2-3 periods, you will learn about the cell division. Then you will come to know about the function of the spindle fiber, about the function of the centrosome. Now this is the brain of the cell or the nucleus. An ideal eukaryotic cell have 4 parts within the nucleus. One is the membrane, that is called nuclear membrane. Another one is a dense structure, it is called nucleolus. One very coiled structure you can see, that is the chromatin fiber, and the gel or semi-liquid structure is present within which the chromatin fibers are present. So that matrix or the base of the nucleus is called nucleoplasm. Now in the time of the division, when the cell will start to divide, you can see outside the nucleus the centrosome. Within the centrosome, 2 parts are there. They will divide and start to make the spindle. And inside the nucleus, you can see a process. It is called condensation. What is that? The fiber-like structures, the thread-like structures, they will condense and will be visible clearly under the microscope. And when they proceed for the cell division, then you can see the nuclear membrane gradually will disappear and all the threads will condense and form a specific structure. It is called the chromosome. You can see in this particular animal cell 4 chromosomes are present, but previously the chromosomes were not seen clearly, but now all 4 separate chromosomes are seen clearly under the electron microscope or high power microscope. So they are nothing but the chromosomes. Now in this class you will learn something about the chromosomes. So this is for your idea where actually the chromosomes present. The chromosomes are the material which carry the hereditary things, means which is transported from our parents, from our ancestors to the next generation. Those genes means carry the materials which carry the hereditary characters from one generation to the another generation is carried by this chromosome only. Here this is the definition of the chromosome. Chromosomes are highly coiled and condensed chromatin fibers. You can see in the diagram condensed chromatin fibers. Now each chromosome contains long DNA molecule. You will learn later on what is the DNA molecule. Full form is deoxyribonucleic acid, and also the protein materials present. Now what is the definition of the chromatin? The chromatin are the thin structure, coil-like structures, that is nothing but the chromatin fibers. So the chromatins are for here. The chromosomes are the chromatids. Both the things are studied by one German scientist, that is Fleming, W. Fleming. Next we will learn about the morphological structure of the chromosome. Now if you see under microscope, under high power microscope, this is a single chromosome. One single chromosome is made up of 2 separate threads. They are called chromatids. So this is a single chromatid, this is another chromatid. 2 chromatids of a same chromosome, they are called sister chromatids. So you can see the sister chromatids are present here within a single chromosome. Now here one constriction you can see where both the chromatids are joining each other. So this constriction is called primary constriction or centromere. This is a vital place where both the chromatids are connected with each other, and when you will learn the cell division chapter, you will learn the chromatids or the chromosomes, they are connected with the spindle in this space only. At the end of the chromosome, this region is called telomere. And here you can see two uneven or sometimes maybe even arms of the chromosome. So this is the short arm and this is the long arm of the chromosome. You will learn later on the types of the chromosomes. Within the chromosome, you can see the thread-like structures. These are nothing but the DNA along with the protein. The chemical structure of the chromosome you will learn in the next class. You can see the chromosomes have taken the part of the chromosomes or the chromatin. They have taken dark color. They are called the heterochromatin. The portions which have taken the light colors, they are called euchromatin. So the lightly stained portion which is dense coil of the chromatid or the chromatin is called euchromatin, and the densely coiled or darkly stained portion of the chromatid is called heterochromatid. Now we will learn the types of the chromosome. We can divide the chromosomes in two separate types depending on the position of the centromere and depending on the staining capacity. Just now what I told, the portion which is taking more stain or which is more condensed is called heterochromatin, and the portion which takes less stained or less coil, that is called euchromatin. And depending on the centromere, you know, this is the centromere of this chromosome, this one is another centromere of the chromosome. So depending on the position of the centromere, we can divide the chromosomes into four separate types. If the centromere is present, one not exactly in the terminal end but very near to the terminal end, then it is called acrocentric chromosome. Under microscope, it looks like J. If the chromosome is more or less in the center but not exactly in the center of the chromosome, then it is called submetacentric chromosome. Under microscope, you can see L shape. If the centromeres are present just in the middle of the chromosome, then this chromosome is called metacentric chromosome. Under microscope, you can see these chromosomes as in the V shape. And last one, when the centromeres are at the end. So another side, the short arms are not visible at all. Only the long arm you can see. Then this is called telocentric chromosome. And under the microscope, you can see these chromosomes just like the letter I. So depending on the positions of the centromere and depending on the position or depending on the staining capacity, we can divide the chromosomes into two separate categories. You have to practice the diagram of these four types of the four types of the chromosome depending on the positions of the centromere. You have to practice this diagram of the chromosome, that is external feature of the chromosomes with all levelings. In your exercise book, you will practice all these things, that flowchart and this diagram with all levelings also. You will practice this three separate cells, diagram of three separate cells showing the condensation of the chromosomes. And at the same time, you will write down these two definitions, and if question comes in ICSE, but definition of the chromosomes and definition of the chromatin, you will write down these two definitions only. Thank you, class.